welcome back to my course, that is aspects of biochemical engineering, and we are now discussing about the kinetics of substrate utilization, product formation and biomass production of microbial cells. Now, in last couple of lectures I tried to discuss that regarding the cell growth kinetics by using Monod and the other equation, which is followed by analysis of the batch process, then CSTR or chemostat and the plug flow reactor. Now, what we observe that chemostat may be the better process as compared to other process. The reason is that it is easy to operate and your productivity also it is quite high as compared to other processes. Now, when and chemostat is such a process, we can maintain a particular phase of growth for infinite period of time. Now, when we discuss about the chemostat, the major drawback of the chemostat is the cell mass that wasting from the reactor. So, cell mass that is going out from the reactor, if it is more as compared to cell mass that is growing in the reactor, then a time will come there will be no cell present in the reactor, what we call de-washout, or in other way, if the hydraulic retention time is less than the generation time, before the cell multiply, you are taking out the cell from the reactor and we meeting the situation of cell washout. And when there is no cell present in the reactor, so there is no reaction take place inside the reactor. Now, to overcome this problem, one approach that we have seen, that is the cell mass recycling. Now, besides the cell mass recycling, there is other technique that also we can use, that is called immobilization of the whole cell. What we can do? We can hold the cell on the solid matrix and then we pass our substrate. Then we will find that that no cell, most of the cell that retain inside the reactor. So, we can easily overcome the cell washout problem. Now, if you look at this, so what do we mean by whole cell immobilization? Now, whole cell immobilization is an alternative to the enzyme immobilization, and is a well developed method for the utilization of enzyme inside the cell from the microbes. So, that in case of this microbial cells, what is it? They have the metabolic pathways. When we immobilize the cell, then the substrate that go inside the cell and undergo the metabolic pathway and give the respective products and take it out. The immobilization of the whole cell become particularly effective when the individual cells become inactive during the direct immobilization technique. So, what I want to mean, suppose this is the solid matrix and we know the active site is this one. Now, by chance this active site of the enzyme is fixed on the solid matrix, then the enzyme will be inactive. Now, here in case of the organism, this is not happen, because this is the cell and inside the cell we have the biomolecules. So, there is no such inactivation is possible. So, when the individual enzyme become inactive during the direct immobilization or the isolation and purification of enzyme is not cost effective, because we always find that proteins are costly. That all proteins are not enzyme. The proteins with the active site, they are enzyme. So, enzymes are more costlier. So, if you consider any kind of pure enzyme, obviously it will be very costly, but when you consider any kind of cell, we will find that it is less costly. And the greatest advantage of the whole cell immobilization is the enzymes inside the whole cell will be active and stable for longer period of time. That is what is not possible in case of this enzymes. Though you know that you can keep the organism active for longer period of time. So, this is the advantage that we have in this particular process. Now, what are the other advantages of this of the whole cell immobilization? If you look into, the multiple enzymes can be introduced in a single step. So, you know that I told you inside the cell we have metabolic pathway. In the metabolic pathways, that a number of steps are involved. So, A to B, B to C, C to C. So, you know that like this, A to B, B to C, C to D. Like this is a number of processes there, and every step you have enzymes, you know, you required enzymes. So, what we consider, this is a multi-enzyme system. So, multi-enzyme system we can introduce as a single step. That is the major advantage of this particular whole cell immobilization system. And if you look at this extraction and purification of the cells are not required, because you can use the cell directly, you do not have to purify it, only the organism that you are supposed to use, that you have to immobilize on the system. Cells are stable for longer period of time. This is the one of the advantage I mentioned before, and it is a cost effective method. I can give a typical example, that Bacillus coagulans, Bacillus this Bacillus coagulant is, it has the glucose isomerase enzyme, glucose isomerase. So, if suppose we have a column, in this column we immobilize this Bacillus coagulant and pass the glucose this one end, and other end we can produce the fructose. So, so obviously it is a cost effective method. We do not have to purify the enzymes. The immobilized whole cell reactor can be operated at a dilution rate that is higher than the maximum specific growth rate of the organism. I told you, since you are holding the cells, you are not allowing the cell to go out of the system. So, dilution rate has little role to play, because dilution, even you cross this D washout, that in the normal chemostat process, that problem can be overcome here. So, this is the major advantage of this whole cell immobilization process. Now, purpose of whole cell immobilization is increase the volumetric productivity, this is one, then increase the product concentration in the outlet stream and decrease the substrate concentration in the outlet stream. This is very important. So, increase the volumetric productivity. What do you mean by volumetric productivity? Volumetric productivity is the amount of product form per unit volume. Now, if we have more volumetric productivity, our recovery cost will be very less, which is more desirable. And product concentration, that that also very important, that if product concentration is high, then also cost of recovery also very high. Then first is the volume. So, if we assume suppose 5 kg per cubic meter of product, and if you increase the volume 100 cubic meter, so how much volume of product, amount of product you can have? 5 into 100, that is 50 kg, am I right? Now, 500 kg. Now, if he has 200, then this will 5 into 200 will be 1000 kg. So, as we increase the volume of the liquid, that your productivity increases, and decrease substrate concentration in the upstream, that the outlet stream is very important. If the concentration in the outlet stream is less, that means what you have as your load for the waste of the water treatment process will be very less, which is that will help to reduce the product cost to a great extent. Now, question comes, what are the methods of whole cell immobilization? We have adsorption, then covalent bonding, then cell to cell cross linking, encapsulation and entrapment. Now, what do you mean by adsorption? We told you the adsorption is a physical phenomenon. Physical phenomenon means this cell is simply adhered on the surface of the solid matrix. So, so the bonding between the cell and the solid matrix is due to the Van der Waals type of holes. So, if you pass your liquid at the very high flow rate, then there will be their axial shear force. Due to presence of the axial shear force, there is every possibility cell may dislodge from the surface of the solid matrix. So, that is the major disadvantage of the adsorption process, but it is considered as the cheapest process because you just pack the material on the solid matrix and pass the cell through the solid matrix so that it can absorb. But when you consider the covalent binding, it is appear to be the very strong binding. This is like this. So, I told you covalent binding is the kind of electron sharing where the electrons we share, that bond is very strong. Cell to cell cross linking, that means I told you this cell with the help of cross linking is like glutaraldehyde, we can have the cross linking, and inside maybe you have solid matrix where maybe that embedded on the surface of the solid matrix. This bonding also covalent bonding, this also very strong bonding. The encapsulation means I told you that day to day life we take lot of capsule, and in the capsule you know that we put the medicine and it is enclave inside the capsule, like we know the envelope, inside the envelope we put the letter. Similarly, inside that coating we put the whole cell. And entrapment means I told you the fiber entrapment, gel entrapment. We have gel, that inside the gel the organism might be entrapped. Now, inside the fiber this organism might be entrapped. So, these are the different immobilization techniques that we have. Now, let us give you some application of this. Now, when you talk about the adsorption process, supporting solid matrix is gelatin, porous glass, the cotton fiber and DE cellulose. So, these are the different solid matrix we can use, and the cells, the one is lactobacillus which convert the lactose to lactic acid. I told you that that lactic acid is very good the raw material, that chemicals for the preservation of different food products. I have given the example of the cheese is a food product, and which can preserve the milk, protein and fat for longer period of time due to the presence of this lactic acid. Now, in case of porous glass, if we use the Saccharomyces cerevisiae, then it convert the glucose to alcohol. Zymomonas mobilis also can convert glucose to ethanol, and Nocordia, it can undergo some kind of steroid transformation process. Now, covalent binding, we have cellulose, cyanuric this chloride, we have Saccharomyces cerevisiae, glucose to ethanol, it can be used. The cross linking, we use the glutaraldehyde is used as a cross linking agent, and E. coli, when you immobilize, then it produces the fumaric acid. Now, then entrapment, we have aluminum alginate, calcium alginate, we have Candida tropicalis, we have Saccharomyces cerevisiae. The one is Candida tropicalis used for phenol degradation, and Saccharomyces cerevisiae convert glucose to ethanol. Then encapsulation, we have polyester, alginate, lysine, this hydroisomer cell, and Streptomyces species. One is used for glucose to fructose, another used for monoclonal antibody production. Now, there are other some examples that I have given here, that is the amino acid synthesis. Fumaric acid, when immobilized on E. coli at 37 degrees centigrade, it converted to L-aspartic acid. 95% conversion of fumaric acid was observed at a flow rate of 0.8 milliliter per milliliter of bed volume per hour. Cell is usually entrapped in the polyacrylamide gel. Now, another industrial application is that raffinose, that is carbohydrate transformation, that when raffinose, it is raffinose is in presence of Motiarella, that is species Venacea, this converted to raffinose to sucrose and galactose. Now, organic acid ethanol in presence of bacterium species, that is converted to acetic acid. Ethanol is converted to acetic acid. Now, another very interesting application that we have, that is the wastewater treatment process. Particularly, I personally involved that in IIT Delhi, that conversion of distillery effluent to methane and carbon dioxide. So, what we have done, we have, it is a basically two-stage process. One is acidogens, another is methanogens. We immobilize the cell in the polypropylene chloride, that solid matrix, and I shall show you how is the solid matrix. Then I shall also show you after the immobilization that how the solid matrix look. So, that we just fix the solid matrix and pass the that distillery effluent, and we continuously produce methane and carbon dioxide, and this process we can operate continuously for 4 months. After that, the performance of the reactor decreases. So, we can stop the reactor and regenerate the system again. So, this is like this. The distillery effluent initially, if you look at the BOD is very high. Initially BOD was 40,000, and this is converted to volatile fatty acid like acetic acid, propionic acid, butyric acid, and this acid converted to methane and carbon dioxide. And so, 40,000 BOD5, that will be converted the 4,000, the 90% removal of the BOD that has been taking place. Now, if you see the experimental setup, it is like this. This is what this is, I call this is the acetogenic reactor, and this is the methanogenic reactor. So, this is acetogenic and this is methanogenic. So, what is happening that here with the help of pump, that this is the we put the we take the distillery effluent here, and we pass the this is the solid matrix inside, we trickle down, and in process is converted to organic acid. Then with the help of pump, one pump is there, we pump this here, and when we pass here, then this acid will convert it to methane and carbon dioxide, and this is the gas collector where we collect the gas. Now, as I told you that if you look at the solid matrix, the polypropylene chloride looks like this. We cut it into, this is 1 inch diameter, we cut it 1 inch by 1 inch, that we cut it into different size, this size, then we pack it in the solid matrix. So, it looks like this, that in the solid matrix we pack this material, and then we prepared the cell suspension, acetogenic culture or methanogenic culture. We pass it like this, and continuously we do until unless we find the cell concentration here constant. Cell concentration is constant. Then we hold it for some time, that with the media, and then let the reaction take place. When your gas production is maximum, then it is replaced by the distillery effluent. So, we continuously pass and produce the acid, and this acid we similarly use for the second reactor for converting acid to methane. Now, this is before immobilization, the solid matrix looks like this. After the immobilization, you can see this is how the cell that attached with the surface of the solid matrix. Now, when you take out the this cell out, it is becoming black. This black color might be due to the H2S that formed during the anaerobic digestion process. This reacts with the polypropylene and it changes the color of the solid matrix. So, another very interesting thing I want to share with you, that is the rotating disc biological contactor. Rotating this biological contactor is a simple rotating disc with a fixed film and biological reactor, which makes use of the microbial cell attached to a specific surface, and microorganism growing attached to the rotating disc transform the soluble organic matter into energy and new cell. This is used for the treatment of organic wastewater. Now, let me explain that, because that suppose I have some picture, because before I show you the picture, let me give you a simple diagram. Suppose this is a rotating shaft. This is the rotating shaft. Now, here we can have the bearing. So, we can have the bearing, and here we can have a motor. So, here the discs are embedded on the surface of the solid matrix. Discs are embedded, and here you put the liquid wastewater in, and this is wastewater out. This is in and this is out. So, it is rotating at a very low RPM. When it rotates, the disc that this is circular disc, when immersed in the water, so it will touch the surface of the liquid. So, it has the weighted surface. In the weighted surface, the cell will be immobilized on the surface, and the organic material that present in the liquid, that will be utilized by this cell and produce the cell mass and other compounds, and carbon dioxide and other compounds. So, some kind of degradation of the organic matter will take place. Now, if you see the picture how it occurs, it is like this. So, that it is with the help of peristaltic pump, you can put the liquid like this, and this is the disc, and this is rotating with the motor, this disc, and here it is rotated like this. So, the cells are that grow on the surface of the solid matrix. This is the disc, and this is how cells are growing on the solid matrix. So, in the outside layer, they will come contact with the liquid, and there we call the aerobic layer, and inside we call the anaerobic layer. So, the inside organisms can carry out some kind of anaerobic reaction, and outside we carried out some kind of aerobic reaction. So, this is, we call it biocoating, and then we take the liquid out in this way. We take in, and we are taking the effluent out in this way. So, in that way we reduce the organic content of the wastewater to a great extent. Now, if you look at this process can be represented in another very simple way. This is the disc, how they mounted on a particular shaft and immersed on a trough, and this is the primary settling tank, and after from this primary settling tank, the supernatant you passes through this rotating this biological contactor, and the reaction take place, and the S0 will be converted to S, and this is the secondary settling tank. Then this is the treated wastewater that we have. Now, this here we can do the substrate balance under steady state condition. What is the rate of input? That is F into S0. Am I right? And what is generation? Generation will be 0. And what is the output? F into S. And what is the reaction that we have? Minus RS, that is the rate of substrate utilization per unit surface area. It depends on the, because cells we assume it presents in the disc, not in the suspension. So, it depends on the area. So, the AS is the area of the weighted surface. So, your RS can be represented like this, where AS is considered as the weighted surface area. Now, considering the Monod equation or Michaelis-Menten equation, we can write in the similar way that that Monod equation minus RS equal to Q max into X into S Ks plus S, where Q max is the maximum specific substrate removal rate. So, this is multiplied by X, then it will be maximum substrate removal rate, and into S Ks plus S. This how we can write this equation. Now, we can write that BPS equation, that that this equation now we can write in this form, that F by A. Now, here we have F, and we have here you have F and A. Now, we can this is equal to this, am I right? This is equal to this, we can write there. Now, and then we can write this equation F by S equal to Q max into X into S, Ks plus S, S and S0. F is the volumetric flow rate. So, then we can write the equation in this form, that is the S equal to like this, and total surface area, this n number of disks there. So, we can write this a generalized equation F into Ks S plus Si, Si minus 1 minus Si Q max by Si. So, this word I stands for 1, 2, 3, 4 to n. This is how we can find out total weighted area, because this weighted area are responsible for carrying out the reaction. Now, another industrial application is that that is a kind of beer industry that we have. Now, in the beer industry, what we have this is the wort. Now, what is the wort? Wort is the fermentation media, and this is when this is Saccharomyces culvergensis. This is used for the mostly for lager beer formation, and this is when it immobilizes on Kieselguhr good or PVC fragments, and then it continuously pass through this column, and we can produce the beer. This is we can parts of oat, and here we will get the beer. That is what is, and then again we are passing is Saccharomyces culvergensis another column to get the mature beer. Whatever unconverted substrate is there, glucose is there, that will be further converted to alcohol. So, the support material was packed with a column 2 meter long and 0.2 meter in diameter. The wort passed through a column at the rate of 3 liter per hour. The column operated 3 months without contamination. So, this is one of the application that we have of this wholesale immobilized system. Now, activity of the wholesale immobilization system can be expressed as the relative activity, comparing the activity of the immobilized cell with the same number of free cells. Suppose we have in the suspension we have cell, and in the immobilized system we have immobilized cells. Now, if a number of cells is same, and if we compare that what is the performance of this suspended cell with respect to immobilized cells, that we call relative activity. Absolute specific activity is the reaction based on the unit weight or unit volume of the whole catalyst. That means per unit cell mass, the immobilized cell mass, that what is the activity, what is the rate of substrate removal, that is actually that called absolute specific activity of the cell. In this connection, I want to tell you that in case of immobilized enzyme with specific activity, we express as the micromoles of substrate converted per minute per gram of solid matrix. Though this is something similar to that. Now, activity of immobilized whole cell, the adsorption and entrapment methods gives R1 value close to 100%, because we assume since the cells are loosely connected on the surface and your diffusion problem is very less, we can expect that 100% of reaction, while the R2 in case of the former where cell are loading inside the cell, then we have some problem, because we have the rate of reaction will be little bit less as compared to the adsorption technique or in the suspended cells. Now, so in case of spore immobilization, let me tell you, in case of filamentous organism, that we have seen that it is difficult, that whether breakage due to the breakage of this this hypha and fragmentation of mycelia, again caused the reducing the activity, but commercial plant exploited the Spore immobilization technique are in operation for the steroid transformation, as fusarium, the fusarium is the use, and this is largely used for the steroid transformation process. So, this is another problem that we have in case of fungal cell. Now, factors that affect the immobilization technique, there are several. One is cell metric interaction, mass transfer and light. These are the different factors that influence this process. Now, if you look at cell metric interaction, when using the reticulous polyurethane foam, this polyurethane foam, we know this is largely used for insulation purpose. In order to immobilize any cell to function well, the volumetric fraction of the foam has to be sufficient enough, so that all the cells and the reticulous pores of the foam is large enough to contain the cells. So, I told you during the immobilization enzyme that when you have pores of the solid matrix, the size of the spore, it should be double the size of the enzyme, otherwise the enzyme cannot enter into the inside the pore. Now, if the pore size is more, then there is every possibility of leaking of the pore. So, pore size plays very important role as per the immobilization of the whole cell. Now, mass transfer, as you know that mass transfer, I told you previously also that as soon as you immobilize the cell, the mass transfer limitation problem that will take place, the substrate has to diffuse, since it is containing two phases, liquid and solid, the substrate to diffuse from the liquid surface to solid surface, and then when the product formation takes place, product has to diffuse from the surface to the. So, that kind of problem that we have, that has been, I am not going in details, you can just go through that. And the mass transfer resistance that has been decreases the nutrient transport, that if your mass transfer resistance is more, the nutrient transfer will be reduced, your growth of the organism that will be reduced to a great extent. Now, light also plays very typical role in case of immobilized whole cell system, particularly photosynthetic organisms, they require light. So, it gives some kind of barrier to the light penetration. So, we shall have to put the LED light inside the reactor, so that light transfer, that transmission may be proper. In that way, we can reduce the light penetration problem to some extent. So, what I wanted to tell in this particular lecture, that how the immobilized whole cell system can be used for in the biochemical industries. It can be used for the production of certain product. It can use for carrying out the series of reaction in a particular one cell. It can use for carrying out a single stage reaction in the by using a particular organism. I have given the example of bacillus coagulant, how it is converted glucose to fructose. I also told you how in the biomethanation process, how distillery effluent can be converted to methane and carbon dioxide. This is a multi-step process. Both acidogens and methanogens, how they involve, and we can get the methane and carbon dioxide. Then I have given you the example of beer formation process. Then what are the different physical parameters, what are the different parameters that influence the immobilized whole cell system, that also we discussed, and the activity of the whole activity of the whole cell, how is affected by this mass transfer, light penetration that I discussed. Thank you very much.